Welcome. So, we have been discussing some of the statistical methods applied to discrimination studies. We discussed about primarily Fisher information and minimum variance unbiased estimator discussions and the relation between the the minimum variance unbiased estimator and Fisher information, which is the Kramer-Rao bound. With that, we discussed on fidelity of coding by neurons based on this Fisher information, and it is a function of the stimulus, and the Fisher information shows how the discrimination performance would be near that very value of S or the stimulus, and that is if we change the S in a very small amount, then how well can we discriminate them. So, as we had discussed in mutual information early on and in some other discussions, we were considering that the stimulus is not necessarily going to be that parametric and that there will be a very small difference, but there can be many different entities as we did for the confusion matrix. Now, we will take up an example where we will try to relate discrimination of two stimuli and try to relate it to performance by a subject. In this case it is a trained monkey that is doing the discrimination task, and the neurons in its cortical region that is thought to be involved in processing such stimuli. The performance based on the neurons will be compared with the actual performance of the monkey and see how well it matches, and at the end we will have a small discussion about the perturbation studies where more about the causality can be established. So, this is work by Britton et al. It is now about 30 years old, 1992. So, in this case, the area of the brain that has been studied is the area called MT, which is in the where visual pathway. If you recollect from our early discussions on the different sensory systems, we had that in the visual system, after the initial stages in the cortical region, the visual pathway is divided into two parts, one that processes what information and goes on to do object recognition, and the other that goes on to process the location or position information and goes on to the more on the dorsal side of the cortex. Now, this area MT is known to be, the neurons in there are known to be direction selective. That is, within the receptive fields of neurons, that is, if we have somewhere the visual field like this, if this represents the entire visual field, then actually the receptive fields are quite large, and if there is motion of a visual object in there, then the neurons here respond strongly to one particular direction and less strongly in the other directions, with a minimal response or even suppressed response in the direction opposite to its preferred direction. So, if we have object movement in the field of view along different directions, this theta varies from 0 to 2 pi or 360 degrees, then if we plot the response rates, then this is our angle theta, direction of motion, and this is rate. Then neurons have a preferred direction, that is some theta naught to which the response rate is maximum, and in the exactly opposite direction, that is theta naught plus pi, it has sort of a minimal response. So this is the opposite direction, theta naught plus pi. In fact, there may not be responses, and sometimes it may even dip below its spontaneous level. So we have now the response characteristics of the neurons that we will be discussing, that they are selective to direction of motion. Now, as we have said all along, these responses are stochastic in nature and generally follow a Poisson-like distribution in terms of spike counts. So, this curve that we have plotted, this tuning to movement direction, that is based on average of many repetitions. So, in general, if I take a particular direction, be it the preferred direction, which is theta naught, there is a large degree of variability in responses. It depends on the kind of neuron and so on. There is a variability around that region. So, that means the rates can be scattered around this region and the average is like this. So, now if we take the histogram of the rates at a particular direction, then let us say this is rate and this is percentage of trials. So, let us say if we present it 20, 30 or 50 or 100 times that moving stimulus in that theta naught direction, then we can get different possible firing rates, and that may have a distribution which is something like this, where this is our mean rate. And this is being, so if this is the response distribution for the angle theta naught, this mean rate is what is being plotted here. So, this rate, mean rate, is what is the mean rate here in the tuning curve. So, we have a spread around that mean rate. So, if we are tasked with the job of discriminating two different directions, then we will, let us say, if it is theta prime here, theta naught, or let us say theta 1, these two, there is a distribution of rates around that also, and so that rate may be here and there may be a distribution of responses around it. So, as we have seen, the task is then to make the decision about whether the two stimuli are different or not depends on the actual spread of these rates for that neuron or the population of neurons. So, we will currently limit the discussion to a single neuron, but can easily be extended to population of neurons. So, given this spread, we can have an overlapping region, and so obviously the more separated with lesser spread these distributions are, we will get better performance in terms of discriminating two different directions of motion. So, this here we are considering theta naught as a continuous sort of variable, just as we discussed in Fisher information. In the example that we will talk about, it will be about specific two directions only, which is the neuron's preferred direction theta naught and the orthogonal the opposite direction theta naught plus pi modulo 2 pi. So, now so since this is an experiment with monkeys, the task was made in this manner to simulate direction of motion, a random dot pattern, a random dot pattern stimulus was used. So, what is this random dot pattern? So, in the receptive field of the neuron, within that region, there are a number of dots at random positions within that region, and so the dots all can move in one particular direction, all of them. So, that move as in the image or the video in every frame is updated in this manner, that is every 45 milliseconds. So, the image that is shown here with the random dots are present for 45 milliseconds, and immediately after that there is an updated position of the dots, which is a small movement in a particular direction. So, all of them shift in a particular direction. Like this. So, the new pattern is by this, and there may be some new dots coming into the field of view or in the receptive field to have a continuity. So, if you can now imagine that in the next instant they move again in that same direction, that would mean that there is a motion in the field of view of the monkey, and so this will clearly have the effect of a motion within the receptive field. And so, the direction tuning, or rather the movement direction tuning of these neurons are obtained. So, we have the theta naught and the theta naught plus pi modulo 2 pi. Anyway, we can drop that modulo. Basically, you understand that preferred direction and the direction opposite to it. So, in this case, when all the dots are moving in the same direction together, it is and all the dots are moving in the opposite direction together, the monkey, which has the task to discriminate these two, can easily do the task. So, what we mean in this case is that when all the dots are moving in sync, all dots are moving in sync, that is that is just as we talked about, all of them are moving in the same direction together. That is what we call a 100% coherence stimulus, 100% coherence, and obviously the task is easy. Now, if we reduce the coherence, that is the percentage of dots that will move in the same direction while the others move in a random direction, then as we reduce this coherence to 30 to 20 and in fact to 1 and then to 0, which means the 0 case is that there is no direction of motion actually, that is the dots are updated in totally random directions every time. So, theta naught and theta naught plus pi have no meaning, and so it is impossible impossible for the monkey in a 2 alternative force choice to do anything correctly. So, it will 50% of the time it will be correct, 50% of the time the monkey would be wrong in terms of detecting this or discriminating the two directions. Two directions in this case has actually is a misnomer, but it is two stimuli, as if there could have been two directions. So, now if we look at the performance of the monkey for the different coherences. So, if this axis is the percentage coherence, as we move to the right, the task gets easy, and to the left, the task is difficult. So, as we said, when the coherence is 0 or extremely small, let us say we will have this logarithmically, let us say it is 0.01%, and then we have a 0.1%, then we have a 1%, then we have a 10%, and of course a 100%. So, this we are plotting logarithmically, and this side is the percentage of times the monkey is correct in discriminating the two directions of motion in its which is shown in that field of view, and the direction is set such that the neuron that they are recording from, that neuron's best direction is theta naught, and its opposite direction. So, the performance would start out, as we said, at a 50% level, that is the monkey on average is basically randomly choosing one of the two, and over a long period of time it will all average out. It is expected to average out to a percentage correct of 50 or 0.5. So, if we are saying percentage, let us correct this and make it 50. And of course, when the monkey is performing perfectly, that is 100% correct. So, what the result showed that means that from this end to the left to the right, it is supposed to go up to 100%, and there would be a position where the monkey will start to discriminate the direction correctly in, that is, and perform above chance level at some intermediate level and reach some the half of the correct, that is 75% or some other criteria 80% at some particular percentage of coherence. What is found is that essentially at up to around 1%, the monkey is unable to detect any of the directions correctly, and within this period from right after 10%, it starts to discriminate perfectly. So, this function, the percentage correct, as a function of percentage coherence, this is the behavioral or psychometric function, that is, this is what the monkey is actually doing in terms of its behavior, how well it is performing for the different coherence levels. So, now in order to actually perform the analysis, if we consider the responses at different coherences, what is observed is that for a 100% coherence stimulus, the rates in the direction of theta naught and the opposite direction, theta naught plus pi, these are the two stimuli. The distribution of rates in the theta naught case is quite high, in the sense that this is rate and this is fraction or percentage of trials. So, what are we plotting here? We are plotting distribution estimated from the response rates with multiple repetitions of the stimulus theta naught. So, let us say 20 or 50 or 100, whatever was used. Based on those number of responses, we have as many firing rate values, and from that we get this distribution. And similarly, for the baseline, similarly for the theta naught plus pi case, we will have another distribution which is going to be very well separated when this is 100% discrimination, 100% coherence. So, this is for 100% coherence. Similarly, as we go to 0% coherence, we will have response distribution. The red one would be similar, and in this case the green one would be also basically the same. I am not overlapping them, basically going to be the same, because there is no apparent direction of motion. And for the other coherences, we will have intermediate sort of overlaps until the two get fully separated, or almost largely separated. So, this is the same axis here, and this is again the same axis rate. So, now based on these rates or rate responses of neurons, assuming that our response measure that is being used by the monkey to do this task is rate, we need to find out how based on those rates we ourselves can or statistically we are able to do this discrimination, and what is the percentage of times we would be correct based on these neural responses. So, in that case, let us consider now there are two stimuli, and that is, we will consider the movement along theta naught as the plus stimulus and movement along the opposite direction as the minus stimulus. So, these are the two stimuli we have, and let us say this is the case of correct and this is the case of incorrect. So, we essentially have something like the confusion matrix that we had discussed earlier, and so we will call this box here is the hit rate, that is, we will represent by the symbol beta. What we mean by that is what is the probability of correct if the stimulus is a plus stimulus, that is probability correct given plus. And we will call, so obviously the probability of incorrect is going to be 1 minus beta, that is probability of incorrect given the plus stimulus. That is, now when the stimulus given is minus, if the probability of incorrect when the stimulus is minus, that means we are saying, based on the responses, we are saying that there is a plus stimulus although there is a minus stimulus. That is what we mean by incorrect with a minus stimulus, and that is called the false alarm. That is, we are falsely saying that the positive stimulus has occurred. So, this will be represented by alpha, and obviously this is going to be 1 minus alpha given the different stimuli. Now, if we assume that these stimuli have equal probability of occurrence, this is half and half, we will get a joint distribution by simply dividing each of these values by 2. So, this is going to be our beta, which is hit, and probability of incorrect given the minor stimulus is our alpha. So, what are we basing our decisions on? That we take the call that it is a plus stimulus based on the responses, or we take the call it is a minus stimulus based on the responses. We have to define something called a threshold here, and that threshold would be somewhere in between the overlapping region, and we can call that Z. So, we have rate distribution here. We will consider it continuous, as we have always said the spike count is discrete, but if we randomize the duration over which we are calculating the rate, then it also becomes a continuous distribution. So, probability of correct given plus is beta is going to be a function of this threshold. That is, if we choose that threshold to be Z, if the rates are larger than Z, then we call that the stimulus is plus. If we have rates that is less than Z, then we call the stimulus is minus. So, if this is our criteria for decision making, then these betas and alphas are going to be a function of Z depending on where I put this threshold. The threshold can be put here, the threshold may be put here, depending on that. So, this is Z, this is Z, and we can also then bring in the rate as the random variable, that is beta Z is probability that our rate is greater than equal to Z given a plus stimulus, and similarly alpha Z is going to be the probability that R is greater than equal to Z given a minus stimulus. So, this is hit and this is false alarm. So, we will now see how to analyze this is to use what we call the ROC curve, or receiver operating characteristics. So, in that what we will see is basically a plot of our alpha and beta as a function of Z, alpha as a function of Z. So, alpha and beta are going to be what we have said, the hit and false alarm, and so since they are probabilities, they would range between 0 to 1 and here also 0 to 1. And for varying Z, we will have different kinds of performances or different kinds of alpha and Z, which is what we call the receiver operating characteristics analysis. So, we will stop here in this lecture and we will start off with analyzing the receiver operating characteristics, the ROC curve in the next lecture. Thank you.